In this video, I'm going to discuss about the structure of maltose here in detail. That is a disaccharide, right? I've already discussed about this sucrose in my previous video. So this is a disaccharide, which is a maltose. Yes, disaccharide. That means it's made up of two monosaccharide units. What are those two monosaccharide units? So this on hydrolysis will give you two molecules of glucose here. That means it is made up of two glucose molecules, one maltose molecule, and the linkage, I mean the two monosaccharide units have been linked by means of a glycosidic linkage. Right, but sucrose, I've already discussed that sucrose on hydrolysis will give you one molecule or equimolar concentrations of glucose and fructose. That means it is made up of glucose and fructose, but here the maltose is made up of two molecules of glucose here, right? So what are those two molecules here? Alpha glucose. So alpha D glucose here is linked to another alpha glucose by means of a glycosidic linkage, at which carbon atom C1 of one glucose and the C4 of another glucose. So let me write the Harvard projections of this alpha glucose. So this is an alpha glucose, right? So I'm putting all the numbering carbon atoms 1, 2, 3, 4, 5, and 6 the carbon atom CH2OH. Right, so alpha glucose, I have which one? H on the top, OH on the bottom, right? This is alpha glucose. And at third position, the glucose, you know, just the opposite. And this is a structure of alpha glucose. Another alpha glucose also, I write the same structure here. Right, so this is the Harvard projections I'm writing. So this is alpha glucose. H and OH top and bottom H, OH. This is just the opposite, H, OH, and CH2OH, H. So this is the sixth carbon atom, right? I'm numbering them 1, 2, 3, 4, 5, and 6. So where is the linkage is being formed between the C1 of one glucose and C4 of another glucose? So this should carbon. So the glycosidic linkage is formed by the loss of this water molecule, right? So I'll remove this water molecule and write the linkage. That's it, right? So this is how the glycosidic linkage is being formed in between the two glucose molecules, that is alpha glucose, and where the linkage is being formed between the C1 of one glucose and the C4 of another glucose, that is alpha glucose. Right, this is the structure of maltose here. And when you observe here, see the free aldehyde group. So where is the aldehyde group is present in the open chain structure at the C1 carbon atom, right? So the C1 carbon atom of first glucose is blocked. That means it is involved in the bond formation. But if you look at the second glucose molecule, here the C1 carbon atom is not involved in any bonding. So this is free. So that is why it can reduce, because of this aldehyde group which is present at the C1 carbon atom, it can reduce failing solution and failing solution in tolerance reagent. So it shows reducing properties. So reducing properties are because of the aldehyde group, because of the second glucose molecule, because the C1 of the second glucose molecule is free here. It is not involved in any bonding. So there is an aldehyde group which is present, which is responsible for the reducing properties. So it is a reducing sugar. But sucrose is a non-reducing sugar because the aldehyde group which is responsible for the reducing properties is blocked there, is involved in the glycosidic linkage, but it is free here. That is why it is a reducing sugar. So this is the structure of maltose. If you like it, please do like, share, and subscribe my channel. The whole chapter has been discussed on my YouTube channel, and the details of it has been given in the description box below.